The next section of the textbook looks at carrier-mediated transport, which is a process that requires membrane proteins to move substrates and ions. And these proteins, like enzymes, show specificity for their substance. Carrier-mediated transport can be passive, meaning that no ATP is required, or active, meaning that ATP is used for energy. And carrier-mediated transport can occur in one of two ways. It can be co-transport, which means that it moves two substances in the same direction, or it could be counter-transport, meaning that two substances are moved, but they're moved in opposite directions at the same time. Some examples of carrier-mediated transport are facilitated diffusion and active transport. So facilitated diffusion uses carrier proteins to passively move larger compounds down a concentration gradient. So passively meaning that we're not going to burn through any ATP or spend any energy to accomplish this diffusion, because remember, diffusion is diffusion is diffusion. It doesn't require energy, whether it's facilitated diffusion or regular run-of-the-mill plain diffusion. So what happens is you have a compound that'll bind to a receptor site, some little niche or pocket or trigger on a carrier protein, and then that protein will change its shape, and by doing so will shuttle or move this compound across the membrane. So it's facilitating this diffusion to occur. However, there's a limited number of carriers, and this can be the only limitation on the rate of the diffusion. So however many carriers are available is how fast that diffusion can occur. And so this is a picture of a facilitated diffusion occurring across a plasma membrane. So here's your extracellular fluid. Here's a molecule that is polar, so that means it can't just diffuse across this non-polar membrane. It has to be facilitated across. It has to be chaperoned to the other side with this carrier protein. So it binds to the receptor site in the carrier protein changes shape, and boom, you've got your molecule inside of the cell. So again, we are still working with a concentration gradient. We're moving from high concentration to low concentration. So that's why it doesn't require any sort of energy. Compare that now to active transport. In active transport, you're working against the concentration gradient. So you're moving things from low concentration to high concentration. Okay, so this is going to require energy. So you're going to be burning through ATPs to make this happen. And one of the ways that we perform active transport is by using ion pumps or exchange pumps. Ion pumps are going to be carrying essential ions to one side of the membrane, where exchange pumps are ATP driven, and they're typically a counter transport mechanism. So probably the best example is the sodium-potassium exchange pump, and that's what's pictured here in this figure, where you have, we're using ATP, it's being broken down to ADP. That high energy bond in that last phosphate group is being broken and exergonically releasing its energy, which this sodium-potassium exchange pump uses to move potassium from the outside of the cell into the cell and move sodium to the outside of the cell from the inside. And notice how there's lots of sodium out here and there's very little sodium in here. So we're moving from low concentration to high concentration. This is the opposite of what's happening in diffusion, which is why it costs so much energy. And it's, again, over here with the potassium, there's no potassium out here. There's some in here. So we're moving from low concentration to high concentration. And that's why it costs so much energy. And so the sodium-potassium pump is so handy because it can accomplish both of these transport mechanisms at the same time in an exchange pump. Now, another type of movement across the membrane is vesicular transport. And so this is where you can actually move these really large amount of material in and out of the cell and through these small little membrane sacs called vesicles. And there's two major categories of vesicular transport. There's endocytosis and exocytosis. Okay? And I bet if you break those words down, figure out what they actually mean. Endocytosis, this is since we're moving such large masses, well, such large quantities of substances, is going to require cellular energy. And what it is we're packaging extracellular material into a vesicle at the cell surface and then moving them into the cell. So take that word endocytosis and break it up. Endo means in, cyto means cell, and SIS is the process of. So this is the process of moving things into the cell with a vesicle. And there's three key types of endocytosis that can occur. There's receptor-mediated endocytosis, there's pinocytosis, and phagocytosis. And so the receptor-mediated endocytosis looks something like this. So here's number one. This is where we start. We have these ligands, or these receptors on the surface of the cell, that once they are bound to their receptors, these ligands will trigger an invagination or cave-like formation on the cell surface. And as it does that, it's going to pinch off. And when these two membranes meet, they kind of like glob together. They form together. They glide together. Remember I told you that cellular membrane is a very fluid, very dynamic structure, and is made up of lipids. Think about if you're washing dishes by hand in the sink, and you've got this oily pan that you put into the sink full of water, and all those bubbles of oil float to the surface, and then once they meet, they glob together and make a bigger. It's the same concept. As soon as those two arms on the cell membrane meet, they're going to join and form this vesicle. Okay, so you have this vesicle, and so now you've moved all of these ligands into the inside of the cell, and that's that process of endocytosis. So this is your receptor-mediated endocytosis, and a variety of things can happen from there. In this example, they're showing how a lysosome can fuse with it and start breaking down those ligands and remove them, and then you can actually just shuttle that vesicle back up to the surface and it'll join again with the surface of the cell and just fuse right with the cell receptors, so that those receptors can then bind with more ligands and continue the process of that receptor-mediated endocytosis. Phagocytosis is very similar, except that phago actually means eat. So this is like cell eating. So here again, we're invaginating the surface of the cell. It's forming these pseudopods that wrap around this substance. It's labeled as a foreign object. It looks a lot like a bacterial cell. And it forms this vesicle on the inside of the cell. And so all of this membrane movement and extension and fusing, that is what costs so much energy. And then from there we have lysosomes again fusing with this vesicle to digest that foreign product, and then releasing it back into the extracellular environment. And so that process of releasing the contents of this vesicle back out into the extracellular environment, and that vesicle then fusing and becoming part of the cell membrane again, that process is exocytosis. And so essentially it's the reverse of endocytosis, of moving material out of the cell by a packaging substance in a vesicle that merges with the membrane. And again, this is going to require cell energy, because again, you're moving large substances around in the cell. So that brings us to checkpoint box. What is the difference between active and passive transport processes? See if you can recall that. Even stop and pause the recording if you need to do so. So this next section of the textbook takes a look at some of the key components of a cell, and it's focusing on those cells and structures of cells that can be found in the human body. And so I want to draw your attention to this figure 3-2, which is a depiction of a non-existent cell, but a cell that has many of the combined features that a lot of your very, very specialized cells do have. So all rolled into one imaginative cell. And so we're going to use it to talk about some of the important structures. First of all, the cytoplasm is this general term for any of the material inside of the cell. So cytoplasm, any material on the inside of the cell. And looking at the cell, I can confidently say that really most of the cells are salty sacs of water. The main component of your cell is cytosol, and that contains dissolved nutrients, ions like the sodium and potassium that I mentioned, soluble and insoluble proteins, waste products, with a consistency like very similar to watery maple syrup. Okay, again, because most of the cell is made up of water. Now, inside of the cytoplasm, or suspended in the cytoplasm, we have organelles. So think of the word organ and then add E-L-L-E behind it. Organelles. And just like the organs of your body, organelles perform specific functions that are essential to the normal cell structure, maintenance, metabolism. And some of these organelles are wrapped up or enclosed in a membrane. So these membrane-enclosed organelles include the nucleus, pictured here. We have the mitochondria, which are these little bean-shaped, worm-like structures, mitochondria. Another membrane-enclosed is the endoplasmic reticulum, which is this squiggly line back and forth here. We also have the Golgi apparatus, let's see, which I'm not seeing pictured real clearly on here, but I think I have a picture of later on. The Golgi apparatus. We also have lysosomes and peroxisomes. So the idea is, here we go, peroxisomes, lysosomes. The idea, here's our Golgi. Ha 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 ha. So the idea behind these membrane-enclosed organelles is that membrane can isolate the organelle from the cytosol, or the liquidy, soupy part of the inside of the cell. And so the organelle can manufacture and store secretions or enzymes or even toxins without it being permeated through the entire cell. So it provides a nice little isolation, that membrane. Some of the non-membrane enclosed organelles are like ribosomes, which are pictured here as just little dots scattered throughout and attached to the endoplasmic reticulum. So that's the difference between free ribosomes, those are the ones that are like sprinkled through, and fixed ribosomes are the ones that are attached to this endoplasmic reticulum. Ribosomes mainly function to manufacture proteins. Proteosomes, which are pictured here, kind of like little tiny bean-shaped structures. These proteosomes are organelles that contain protein-breaking enzymes, which are called proteases or proteolytic enzymes. We also have centrioles right here. These little structures, they almost look like noodles. These are cylindrical in shape. They move DNA strands during cell division. So we only talk about those when we're talking about mitosis and meiosis for the most part. And then we also have cilia pictured here. Cilia, which are kind of like little finger-like projections out into the external environment of the cell, and they're going to burn ATP, but they sweep food particles or dust particles. They move substances across the surface of the cell, which is different than flagella. And they don't have any flagella pictured here, but a cell with an example of a flagella would be like a sperm cell. So these are long cilia used to move the cell through its environment instead of moving stuff over the surface of the cell like what cilia do. They look a lot like a tail, and they propel that cell through their environment. We also have the cytoskeleton pictured here. So let's take a closer look at the cytoskeleton. It's an internal protein framework that provides strength and flexibility to the cell. The most important cytoskeleton elements in most cells are microfilaments, which are made up of actin, intermediate filaments, which stabilize the cell's position with respect to surrounding cells, And then microtubules. Microtubules are going to add rigidity and anchor the position of organelles. They'll form the centroles, the cilia, and the flagella. So microtubules are a very important part of the cytoskeleton. And then we also have pictured here, in addition to our microfilaments, and our microtubules right here, and intermediate filaments, we have these microvilli that are pictured. So microvilli are finger-shaped projections that are on exposed surfaces of some cells, and they can provide an extra surface area for that cell. So they're found on cells that transport substances from the external cellular fluid. This next figure is showing a close-up of the endoplasmic reticulum. The endoplasmic reticulum is this network of intercellular membranes that are surrounding the nucleus. And we have two different varieties of endoplasmic reticulum. There's the smooth endoplasmic reticulum, sometimes abbreviated SER, and the rough endoplasmic reticulum, which is sometimes abbreviated RER. The smooth endoplasmic reticulum is called smooth because it doesn't have any ribosomes attached to it. A smooth endoplasmic reticulum is where lipids and carbohydrates are synthesized. The rough endoplasmic reticulum is called rough because it has these fixed ribosomes that are attached to the membrane. This is typically where proteins are synthesized, and that's why they have those ribosomes attached, because that's the purpose of ribosomes, to make proteins. I just want to take a close-up look at the Golgi apparatus. As I said, I had a better picture for you, and here it is. So the Golgi apparatus is made of flattened membranous discs that are called cisternae, and there's three major functions. They modify and package secretions and form secretory vesicles, as what's right here in this picture. They renew or modify the plasma membrane by generating membrane renewal vesicles. Right here. They just kind of pinch it off and it floats out to the membrane, and you've got a new little section of membrane. And then they'll package enzymes as well, and these are called lysosomes. So these lysosomes contain different enzymes that are packaged in the Golgi apparatus. And so these lysosomes, that was another listed structure, another listed organelle. They're produced by the Golgi apparatus, and they contain digestive enzymes. So they help with cleanup and recycling of materials within the cell. They can help with immunity, and they can trigger autolysis, which is sometimes called cell death. Another type of packaged enzyme is the perioxisome, which I don't have pictured here, but was pictured in that 3-2. And so these contain enzymes that will break down fatty acids. So that's a very specific type of enzyme. The mitochondria are a very important organelle for the cell. They are formed by a double membrane, and the outer surrounds the organelle, whereas the inner is folded back and forth and forms what's called a cristae. And the cristae increase the surface area exposed to the fluid matrix on the inside of that mitochondria. And the reason that it has this folded membrane back and forth is because the mitochondria serve as the site of major ATP production through aerobic metabolism or cellular respiration. Probably one of my favorite organelles, and the largest, easiest to be seen with a light microscope, is the nucleus. And so the nucleus is often called the control center of the cell, meaning that it dictates cell function and structure by controlling protein synthesis. The nucleus is separated from the cytosol by a nuclear envelope, so its membrane has a very special name, nuclear envelope, and is a double membrane that surrounds the nucleus and has nuclear pores. So these nuclear pores allow some movement of substances in and out of the nucleus. For example, small single-stranded RNAs can get out, but double-stranded DNA molecules cannot get out of these nuclear pores. So DNA stays in, RNA floats out. Other important structures in the nucleus include the nucleoli, which functions to synthesize that ribosomal RNA, also called rRNA. And the nucleus is the site for chromatin, which is the DNA strands that contain genetic code and dictate the organism's inherited traits. So let's take a look at that. We have chromosomes. Chromosomes, so here's our cell, here's our nucleus, and here's a picture of that chromatin. The chromosomes, as pictured here, are built of DNA and special packaging proteins called histones. You can see the histones pictured here. Histones. So here's your DNA wrapped around the histones. And similarly, chromatin is loosely coiled. So here's chromatin. Chromatin is that loosely coiled DNA found in cells that are not dividing. So do you see the difference? This is a cell that's being prepared for division, which is why chromatin is packaged around the histones, creating chromosomes. Non-dividing cells, they have their DNA all unraveled in chromatin. So the DNA really only takes on the form of a chromosome when the cell is about to divide. And so chromatin is the working form of DNA for the cell. And that chromatin DNA strands contain the genetic code that dictate an organism's inherited traits. The genetic code is arranged in triplets, meaning that 3 nucleotides dictate or code for 1 amino acid. Kind of like how a recipe is a combination of ingredients that form a cookie. So genes are functional units of hereditary that contain all the triplet codes needed to produce specific sequences of amino acids that fold into functional proteins. So I'm wondering if we could pause the recording, or maybe we'll end this recording and start with the next section on the next recording, but take a moment and try and identify the membranous organelles and list their functions.